Hi, Dr. Roshanak. Nice to see you here again. Feel free to come up to the stage. We'll wait a few more minutes for the speaker to arrive, but here's the presentation. Nice to see you here.
Hi.
I'm so glad you could make it.
Oh, I'm so happy too. I just saw it and I thought, oh, this is fantastic. What a wonderful topic to be discussing today.
Yeah, we wanted to focus more on glia because, they were historically so underrepresented in the...
Well, and it's also important when we talk about immune function and then how that also correlates to mental health, right?
Exactly. So, yeah, I'm really looking forward to hearing our guest speaker. She's really nice. I had the introduction meeting with her. She's really amazing and nice scientist. So, yeah, it will be really interesting today.
Yeah, I'm looking forward to it. Let's share the room out.
Yeah, exactly. I'll share the room. Thanks.
Hello.
Hello.
Hi, Sasirahim. How are you today?
I'm fine. How are you?
Good, good. How was your vacation?
Oh, it was so good. So much fun.
I'm so glad to hear that.
Yeah.
It's been a...
Hello.
Hello.
Just saying hi to Dr. Hashimiyun.
Hello, hello.
Did I say that right? Hashimiyun?
Perfectly. Thank you.
Roshanak Hashimiyun, right?
Yes, I'm super excited to hear from you today.
Yeah, I mean, welcome to the team.
Thank you.
Oh, hi, yes, Jessica.
Hello.
Welcome.
Thank you.
Hello, Jessica. How are you?
Good. How are you guys?
Fine, we're fine.
Hello,
Hey, Serena.
how are you?
Everything's good. Steven. Kirko.
Hey, Kirko.
He's probably away from his phone.
So, Dr. Bolton, have you spoken on Clubhouse before? Have you been here before? Is this your first time?
I have not spoken on Clubhouse before.
Well, welcome.
Yeah, thank you.
We're often bringing in some academics, right, or people who are not usually on this app.
Right.
Yeah, and they're like, and it turns out to be quite nice. And I think it's really wonderful that that you're here, that Katerina has brought you here, that we have the opportunity to get some, really important and powerful academic information, because there's so many people that come around and they're just, talking, but to get the really well researched work. And I see we have Girish and Dr. Natalie in the house. I think that's wonderful, and I really appreciate that. Thank you.
Yeah. Thank you.
Well, I've been really excited to hear this talk. I've recently just become obsessed with the neuroglia, so
Oh,
very exciting.
awesome.
I too am becoming very obsessed with them.
They are very cool, I have to
I
say.
slipped and fell down the astrocyte rabbit hole.
Oh, nice. That's a good way to say it.
I'm also interested in the way that they are involved in sleep and
Yes.
rejuvenation, and then how that correlates with mental health, right?
Right. Yeah, there's a lot there. It's all very new, I feel like still, but definitely very interesting.
Well, and particularly the astrocyte syncytium and the calcium waves and how they would differ in the theta region, waking versus sleep. It's just fascinating stuff.
Yeah, that's very true. Definitely.
So we'll wait a couple more minutes. Katerina, you there? Or oh,
Hey, yeah, sorry, my daughter's taking forever. She has a dress down there, you know, they have school uniform, so she's like all excited about picking an outfit still. Sorry. So usually she's in bed by now, but she's very excited.
Oh,
Yeah, so thank you so much, everyone, for coming today. Welcome to Science Society, and a special thanks to Dr. Jessica Bolton, who will talk today about her really amazing neuroscience work, research, and her work focuses on glia. And let me give you a little bit of background information. And then, if it's okay with you, Jessica,
yeah.
one of us will ask you a more general question, and then the stage is yours for the presentation. So,
Okay.
um, Dr. Jessica Bolton, she did her bachelor's in science in animal behavior at the Southwestern University with summa cum laude, and her PhD in 2015 in psychology and neuroscience at Duke University. I was also at Duke University at some point.
Oh,
And
nice.
her, yeah, as a PhD student in George Augustine's lab. I don't know if
Oh,
you're still.
cool. Yeah.
Yeah. And her postdoctoral fellow at the University of California, Irvine.
Hmm.
And her specializations are in neuroimmunology, microglia neuron interactions, early life stress, adversity, neurodevelopment, and behavioral neuroscience. And yeah, we are very honored to have you here today, and we are very excited to hear you speak. But before we start, maybe Cecilium, do you want to ask like the more general question today?
Yes, I'll be happy to. Thank you so much, Dr. Bolton, for coming to see us, to speak to us today. So we
Sure.
just kind of like to ask some question just to get to know you a little bit. So I wanted to ask you, how did you get into science? What was the motivation for that? And kind of how's the career trajectory been? Was it something that you've always wanted to do, or is it something you just maybe stumbled upon? And yeah.
It's a great question. Thank you. I'm very happy to be here. Yeah, that's always something I think about because I don't think I knew what being a researcher was when I was growing up. So, people knew about being a doctor, that kind of thing, if you liked science, but not really research as much. So, at first I just wanted to be a doctor. That was the path, since I knew I loved science and my classes in high school and that kind of thing. I actually grew up loving animals as well, so I learned like all of the names of the baby animals, the different species. I thought for a while I wanted to be working in a zoo or something like that. Then I wanted to be a veterinarian. Then I wanted to be a doctor. So it progressed. And then I took a class in college. I think it was my sophomore year. That was called Research Methods in I think it was in behavioral neuroscience actually, which I fell in love with and realized that's what I wanted to do as soon as I took that class. So I think that was really the start of my more scientific path, or like following the path of scientific research. And from then on, I kind of just went full steam ahead. I went directly to graduate school after college. And, that was kind of a funny story actually, because I did a summer program when I was a sophomore in college with my future graduate advisor, and fell in love with that lab. So I went back for grad school at Duke with Stacy Bilbo. So anyway, that kind of unfolded um, pretty naturally, I guess. And then I went to do my postdoc at UC Irvine with Tally Barham. And yeah, just very recently, I guess about a year now, I started my own lab about a year ago at Georgia State University. So it's all kind of unfolded up until this point.
Thank you so much for sharing that. Yeah, and I totally understand. Like sometimes we don't know how many options there are in the science field until you're actually in it, and you're like, "Oh my goodness, there's so many things
Yeah,
I could do." Yeah. So that's
exactly.
definitely quite wonderful. Yeah.
Yeah, exactly. And yeah, I should have mentioned that that summer program I did in college is where I fell in love with microglia, because I was paired with Stacy Bilbo, who studies microglia. And I realized that's what I wanted to do for grad school. So yeah, they're these cool, alien invaders of the brain, basically, that aren't born there normally. Like the other cell types, like neurons and astrocytes and oligodendrocytes, they just come from the yolk sac during development. And I thought that was so cool. Like, what are they doing there? What's their evolutionary function, basically? So that really got me fascinated starting early on.
Thank you. Thank you so much for sharing that. Yeah.
Sure.
Okay. Well, yeah, I guess we can go into the presentation now.
Alrighty. Let me make sure I'm on the right. Okay. Well, yeah, so I sent some slides ahead of time if you guys wanted to follow along. I realized though that I had some cool videos, but lately realized that the PDF isn't going to show that probably. So I apologize in advance for you not being able to see the cool 2-photon videos that I've put in this presentation, kind of in the wrong format probably. So anyway, you can just imagine. But just to get started here, I'll go ahead and move on to slide 2, and just start with the basic background of why I study what I study. Basically, we know that early life experiences affect your risk or resilience to these mental disorders later in life, including things like depression, addiction, and even dementia, lower cognitive abilities, things like that. And we kind of think about this in my lab as kind of two sides of the spectrum, or two sides of the coin. Kind of having optimal early life experiences leads to more often resilience to these mental disorders, whereas chronic early life adversity and stress on the other side of the coin lead to risk for these same disorders. And of course, all of these things interact with genetics. So it's nature and nurture. It's not one or the other. But for the purposes of my work, we focus mostly on the experience side of the coin, the environment side, and how that really can shape risk and resilience to these mental disorders. So moving on to the next slide here, we'd like to think about the brain, obviously, as organized as these networks called circuits. And the function of these circuits is what underlies complex behaviors, like you would consider making up things like your mental health or your risk or resilience for mental disorders. It's a very complex concept, obviously, but we can try to break it down as neuroscientists into the component parts, right? So we can think about the circuit as a whole. We can also think about the individual components, or the neurons that are making up these circuits. And even more importantly, I think the connections, I guess, between these neurons, or the synapses, I think are critical for the function of these circuits, and especially how things are wired up during development. So if you go down to the next slide, really the question is, do early life experiences influence circuit maturation during development? Mainly, this is important because we know circuits are immature. They're still plastic and malleable early in life. So there's a kind of right situation, right time, right place, essentially, for these experiences to change how your brain's developing early on. And my question coming in as a postdoc to my lab at UC Irvine with Tali Barham was really trying to understand if microglia might play a role in these kind of changes that are happening with early life experiences in your circuits and in the connections between your neurons. And the reason I was really interested in this is because of my passion for microglia that I developed during my PhD work with Stacy Bilbo. And during my time in my PhD, I think that was 2010 to 2015, one of the most seminal papers came out in this field from Beth Stevens' lab at Harvard. And they found that microglia actually do something what we call synaptic pruning. They actually engulf synapses or eat them, quote unquote, because they are immune cells like macrophages in the skin or the periphery of the body. Essentially, they can do similar functions. They can eat things, but not just dead or dying cells. They can also eat synapses and bits of cells, which is really interesting. And so Beth Stevens' lab and Dory Schaefer, as the first author of this paper, showed that microglia really do this important function during development, and specifically during critical periods of development. For example, they found at day 5 in a mouse pup is when there's a high amount of synaptic pruning in this brain region they looked at, called the visual thalamus. Sorry, I apologize for my cat. He always wants attention at inopportune moments, if you can hear him. But we'll see if he gets distracted. So anyway, the sensitive period really varies by brain region, but here in the visual thalamus, it was really about day 5 of life for the mouse. And it goes down the synaptic pruning level pretty dramatically by P9 or postnatal day 9, and 30 days into their lives as well. It's even lower. So now we're moving on to slide 6, and basically I wanted to, in my postdoctoral work, use this naturalistic, relevant model that my postdoc lab had developed called limited bedding and nesting, which is a mouse model of chronic early life adversity or stress. And so Tali Barham had invented this model about 20 years ago, and it's quite simple, really, but pretty strong. So all you do is limit the amount of bedding and nesting material that you give a mother mouse early in life when her pups have just been born. So this is postnatal days 2 to 9 in a mouse. And the mother mouse, with this kind of half amount of bedding that she's used to have, will start to behave differently, actually. So what we see is this fragmented and unpredictable behavior from the mother because of this impoverished environment or limitation of resources, essentially. And long ago, they validated that this model does indeed increase levels of stress hormones, such as corticosterone in the mouse, which is similar to cortisol in humans. And so these levels of increased stress hormones actually persist from being high soon after the paradigm's over to still being high later into adulthood. So it seems like this change has been programmed somewhat into the offspring, which is really interesting. And then going on to slide 7, you can see that we also have behavioral changes that we've explored in this model, not just the stress hormone changes, but we also see vulnerability to later stress. We see things like anhedonia, which is the lack of pleasure in response to everyday things, which is a symptom of depression. Cognitive deficits are a big thing in this model as well, along with progressive cognitive decline. So something like dementia shows up in these mice as well. And then to move on to the next slide, slide 8 here, we are looking at the overview basically of my talk here. Basically the pathway by which we think early life adversity is resulting in susceptibility to stress-related and emotional disorders like anhedonia later in life. And we think this involves microglial synaptic pruning and ultimately the altered epigenetic programming of neuronal populations and stress circuit changes as well. So we'll get more into this in the talk today. As just a little background on slide 9, I have kind of a schematic of how the brain responds to stress. For those of you maybe not familiar, it's basically showing you the HPA axis, or the hypothalamic pituitary adrenal axis, which is kind of the canonical stress response center in your body. So it starts up in the hypothalamus in the brain, which is where you'll see the pink circle there on the slide. That's where corticotropin-releasing hormone, or CRH, is produced in the paraventricular nucleus of the hypothalamus, or the PVN. And we'll be talking about that more throughout the talk today. So just to get you oriented to that central node of the stress response in the brain. And then that molecular mediator CRH is shuttled down to the pituitary, where that pink arrow is going, below the base of the brain. And there ACTH is made, which is transported into the bloodstream and sent down to the adrenal glands on top of your kidneys, which is where the more canonical stress hormones we think about are made. So things like cortisol and corticosterone in rodents are released there. And this is kind of the canonical stress response. I also have in the bottom left corner of this slide some basically an image, a confocal image, fluorescent microscopy image of the PVN in the hypothalamus in a little more detail. So we have the CRH neurons highlighted here in red. That's TD tomato expressing neurons for the cognoscenti, and also green immunolabeling for CRH itself. So just some validation of a transgenic mouse line that we use. That basically is a CRH reporter line in these mice, so we can see these CRH neurons really easily, which is great. So moving on to slide 10, early life experiences, again, this kind of two sides of the coin, have been shown to bidirectionally influence synaptic innervation of CRH neurons in the PVN, this brain region that we've been talking about so far. And so again, on the optimal, or the good side of the coin, with augmented maternal care, so to speak, we have what actually results in reduced excitatory transmission of these CRH neurons. So what I'm showing you in the figure, if you're looking at those slides, is that VGLUT2, a marker of excitatory synapses, is actually decreased in these are rats actually that got augmented or optimal maternal care. So this results in decreased excitation of these CRH neurons, and thus a lower stress response later in life. On the other side of that coin, the chronic early life adversity or stress side, you actually get the opposite, which is really interesting. You get increased excitatory transmission in these CRH neurons. So if you look at the confocal images there, you can see that there's more VGLUT2, now shown in red, around these CRH neurons, which is suggesting that there's increased excitatory transmission in these neurons. And this actually results in more CRH expression, more stress hormone production, and a greater stress response. So you can kind of see how this bidirectional relationship is really coming into play here. And then if you go to the next slide, slide 11, on this one, I'm showing you that stress-sensitive CRH neurons in the PVN are actually programmed long-term by these early life experiences. So in the case of the good side of the coin, the augmented care, oops, that's Siri, excuse me. Basically, we're seeing with the augmented care conditions that there's less CRH production long-term, which is programmed again into adulthood by this early life experience in the PVN. And you also even see in terms of the stress response, if you give these animals a restraint stress later in life, they show a much lower stress response in terms of their stress hormone production compared to control animals. So they're really resilient to these later stresses, which is very interesting. So my question again, coming into the lab in my postdoc was, if these changes in synaptic innervation that are caused by early life experiences are so important, then how are they occurring? So I really wanted to figure out the mechanism by which these changes in synaptic number were taking place after early life experiences. So looking on slide 13 now, I started in the lab just by asking the simple question of whether microglia are actually there in this brain region when we're talking about these early life experiences taking place. So, for example, I looked at postnatal day 4 in a mouse and postnatal day 8, and I compared the number of microglia and neurons in these regions, and yes, they are there even early on, which is not actually very surprising because microglia do, again, enter the brain early during embryonic development. They do kind of spread out into certain brain regions at slower rates than others, so it's always good to look to be sure. But they are here in the PVN even 4 days after birth. And what was interesting is that over time, over even those 4 days, we do see more intimate kind of co-localization or interaction between microglia, which are shown in green on that slide, and the CRH neurons, which are shown in red. And you can see that co-localization in yellow there on the right bottom corner. And that's quantified in the graph, just showing that there's more colocalization between these cell types over time. So moving on to the next one, the next question was, okay, well, they've already shown in recent work, like Beth Stevens' work out of Harvard, that microglia prune synapses in certain brain regions, right? Like the visual thalamus, hippocampus had also been explored, but no one had really looked at the PVN. So I needed to answer that question first to be confident that yes, they do actually prune synapses in this brain region. So the best way to go about that in my head was to inhibit microglial function, specifically their synaptic engulfment, and then ask what changes in terms of the synapses number. So in this experiment, I basically treated microglia with a MerTK inhibitor, which is just a drug that inhibits the phagocytic receptor on microglia that enables them to engulf or prune synapses. So if you inhibit that function and then look at the number of excitatory synapses, you see an increase in the number of synapses, which does suggest that normally microglia are playing a role in synaptic pruning, because if you inhibit them, you get an increase in the number of synapses. And that was interesting because if you go to the next slide, 15, it looks very similar to the increase in number of excitatory synapses that we see after early life adversity or ELA. And so that was an interesting kind of mimic here, in that if you inhibit microglia, you get an increase in excitatory synapses, and ELA also causes that a similar magnitude increase on these CRH neurons in the PVN, both at P10 at the end of that early life experience, and also kind of enduringly, even a couple weeks later, at postnatal day 25 in these mice. That increase is still there in the number of excitatory synapses, which we assume is probably persisting into adulthood. Now, we also looked at, on slide 16, the function of these synapses, not just the structural changes. And we found that early life adversity does augment excitatory signaling in terms of the spontaneous excitatory postsynaptic currents and their frequency. We saw an increased frequency in the ELA animals, whereas we did not see a change in the frequency of the inhibitory signaling, the spontaneous iPSCs. So that really kind of signaled to us that we should focus on the excitatory side of the equation, which have really been shown in previous work as well to be more impacted by this ELA experience. So putting all of that together, my hypothesis on slide 17 is showing that really early life adversity seems to be inhibiting microglial synaptic pruning of CRH neurons to result in that excess of synapses. So that's what I set out to explore further and more mechanistically in my postdoctoral work. And so, of course, the first thing many people look at when they're interested in microglia is, are there more of them? Are there fewer of them? Are they a different morphology? Do they look angry? So we did all of those things and we really didn't find any changes. We didn't see changes in the number of microglia or their morphology. We didn't find changes either in CRH neuron number, suggesting that probably the microglia aren't eating differentially different numbers, I guess, of these CRH neurons during development. It's probably more at the connection level, the synapse level. So from there, looking now on slide 19, we wanted to get more functionally investigative, basically the function of these microglia rather than just kind of the gross level changes that we might be able to see. So the best way I could think of to do that was to do some live 2-photon imaging of these microglia interacting with the PVN CRH neurons in living tissue. So on the slide 20, I guess this would be another video that you probably cannot see playing, but this is essentially an example of the 2-photon imaging that we implemented in acute brain slices of the PVN from postnatal day 8 dual reporter mice. And so they basically have the red signal in their CRH neurons, and they have green fluorescence in their microglia. So it enables us to see those cell types interacting live in living tissue, which is really cool. And so we were imaging these slices as quickly as we could post-sacrifice in order to get kind of as little artifactual kind of microglial activation as possible. But they ended up working out pretty well, which was great, producing these really nice videos. Now, if you go to the next slide, slide 21, we then ran into the issue, though, of, okay, we have these great videos, very pretty, but how do we actually quantify them? How do we get data out of this besides just watching them, right? So what you can do, there are multiple ways to do this, obviously, but one way we picked up early on was a pretty manual analysis approach called a chymograph, which is just a time by distance plot, which allows you to measure the movement of anything, basically, in a video. So we basically were manually tracing the microglial processes and tracking them over time in these videos, over frames, essentially. So we would trace the extent of the microglia's little arm or its process that it's using to reach out and survey its environment, which is something they do normally in vivo in the living animal and in these living slices that we collected. So you can see that over time, you know, in the first row there for cell number 1, the microglial process that's showing in frame 1, it actually retracts or pulls in by frame 30, and you can see that visually in the chymograph where it pulls in over time, over frames towards the cell body. Another example shown in cell 2 on the bottom row, that the process actually grows out and then pulls back in later. So just an example of how that's quantified. But in the next slide is where we actually were able to quantify everything, get the total distance traveled by these microglial processes over time, over these 30-minute videos, and then see that ELA indeed decreases the process distance traveled by these microglia. So, interestingly, that suggests that the function of these microglia seems to be inhibited by ELA, which is matching my hypothesis that maybe ELA is just kind of shutting down their function to some extent or inhibiting them. Now, of course, I didn't believe it with just one approach like this, so I wanted to use another approach and to analyze these videos since they were so complex. So we recruited to my postdoc lab a rotating graduate student who'd come from NASA, actually, Jacqueline Beck. She's really amazing. She created this pipeline in Python, essentially, to automatically skeletonize these microglia in the videos and track the processes in a much more automated way than we had in those chymograph plots. And so what was really cool, though, is that she found a very similar result with this totally independent approach that she used to analyze the same videos. So she found that the process velocity of these microglial processes was diminished in the ELA mice. So again, they seem to be functionally inhibited by this early life experience, which is cool. So now it's more believable. So moving on to slide 24, having it showed or talked to you guys about how they're actually engulfing synapses. So we did do some really cool electron microscopy work with Marie-Ève Tremblay in Canada, and confirmed at the nanoscale essentially that these microglia are contacting and engulfing synaptic elements in the developing PVN, really. So then when we went to quantify this with confocal microscopy in slide 25, in the controls versus ELA animals, we see that early life adversity results in a decrease in synaptic engulfment of microglia, specifically those that are contacting CRH neurons, though, which was really interesting. So, if you look at the top graph on slide 25 on the right, you see a significant decrease in ELA in terms of engulfment. But if you look at the bottom right panel, the microglia that are not contacting CRH neurons actually show no significant difference with ELA, suggesting maybe that these CRH neurons are directly signaling to the microglia and causing this decrease in synapse engulfment of these of the synapses that are on their cells. So that was really interesting. The next question here was really, what's the mechanism of this impaired microglial synapse pruning in the developing PVN? So moving on to slide 27, we can see that really we started looking at this phagocytic receptor MerTK, which I mentioned earlier, and we found that it's expressed primarily in microglia in the immature PVN. We did not see it expressed in astrocytes, which are another very important type of glia in the brain that some people have reported do express MerTK in other brain regions, but we don't really see it in the PVN at this time point. So primarily in microglia, which is interesting for us. And then going on to slide 28, when we compare control and ELA mice, and we look at the amount of MerTK that's there in these microglia, we see a decreased amount of expression of this phagocytic receptor, MerTK, suggesting that maybe it's functionally diminished because there's less of it. There's no difference in the total volume of microglia, the P2RY12 volume that I'm showing you. as well as no difference in the volume of the PVN itself. So it's really this diminished expression of MerTK, the phagocytic receptor, that we're seeing. And then going on to slide 29, we see that if we did a functional experiment in vitro, we basically delivered this MerTK inhibitor to organotypic slice cultures and found that this actually increased excitatory synapses on CRH neurons in control animals or control PVN slices, which is what we did before when I showed you that inhibiting microglia and their synaptic pruning increases synapses. Okay, we did that again. But what was really cool was that if you do this in ELA slices from ELA mice, you don't see a further increase in excitatory synapses, which suggests to us really that this is kind of already a pathway that's inhibited by ELA. So we call this an occlusion effect, essentially, where it can't increase anymore because it's already inhibited, this pathway, the MERTK signaling. So that was really interesting and suggested that this phagocytic pathway in microglia is most likely inhibited by ELA, which is why we're ending up maybe with this decreased amount of synaptic pruning and increased amount of synapses. All right, so the last question here, the last part of the talk is really the question of causation, really. So can we reverse the ELA-induced synapse access that we see by reactivating microglia? And this is kind of a necessary question, like, is this inhibition of microglial function necessary for the synaptic rewiring by ELA? So to get at that question, the approach that I took that seemed the best in this case was using a chemogenetic approach. And if you guys don't know what that means, it's basically using these designer receptors exclusively activated by designer drugs. They're called DREADs. It's a very cool tool, essentially very commonly used in neuroscience these days, but very commonly used in neurons, I should say. It's not very commonly used in microglia. So I applied this tool, this chemogenetic approach, to microglia, and basically used transgenic mouse lines crossed together to do this, and produce litters that essentially had 50% of the litters expressing this GQ or activating type DREAD in their microglia, and 50% of the litters were expressing were not expressing the DREAD because they did not have the CRE, essentially. So 50% have this tool that we can reactivate, quote unquote, microglia during ELA, and 50% are controls, essentially. So this timeline in slide 31 is showing you that basically we insert a pellet with the drug, the designer drug that is part of that acronym, the DREADs that I was talking about. So this is clozapine N oxide, or CNO. That's something that activates the DREADs that we've inserted into these transgenic mice, and shouldn't activate anything else. And so over basically a week from postnatal day 3 to postnatal day 10, while these animals are experiencing ELA, they also are receiving kind of this slow release of drug, of either CNO or placebo, the matrix essentially of this pellet. And so then we can look at their number of excitatory synapses at P10 at the end of that experience, or we can grow those animals up later to look at more functional outcomes such as stress reactivity in adulthood. And so to do that, we actually measured the stress response to an adult stress, looked at their ACTH and corticosterone levels in their blood. We also collected adrenal weights, which is kind of a measure of chronic stress throughout life. And we measured their behavioral response to a predator cue, which is in something called the looming shadow task that we'll talk about in a bit. So that's just an overview of the experimental design. And looking at the next one, slide 32, this is just kind of validation that the approach that we took is working to some extent. So we were able to specifically validate that our dreads were expressed in the microglia, specifically in the developing PVN, which is always good. So the next one, slide 33, we did first kind of a ex vivo validation study looking at, in our acute slices of the PVN, if we're imaging on the 2-photon microscope and we add CNO or vehicle to the media that's perfusing on these slices, basically what happens, what changes with the microglial process dynamics that we can measure. And what we saw is that when you add CNO, this drug that will exclusively activate the GQ dreads, basically we see that the microglia move their processes more, which does agree with the idea that we're kind of activating them with this dread system. So that was a nice kind of validation of the idea, this crazy idea that we were implementing. So moving on to the next slide, 34, we were then looking at the number of excitatory synapses in these mice in the PVN on these CRH neurons. And so the hypothesis shown on the left is that, again, early life adversity inhibits synaptic pruning. So if we give this kind of chemogenetic intervention approach with the dreads, we'll reactivate the microglia and recharge them basically to do their synaptic pruning duty. And so what we found agreed with that hypothesis, that if we look at the far right light pink bar, the ELA group that got the GQ dreads with the drug to reactivate, basically we see a reduction in the number of excitatory synapses back to control levels. Even though these animals are experiencing early life adversity, we can prevent this increase in synapses from happening with this reactivation strategy, which is really cool. Now, the real question, though, is what's the functional outcome of all of that? It's great to see changes in synapse number, microglial motion, process dynamics, but what does that mean for stress reactivity in adulthood? So this is where we looked at these more functional outcomes. So moving on to slide 36, if you look at the kind of response in terms of stress hormones of these animals to an adult stressor, we see that ELA mice have a prolonged stress response, and this is in terms of ACTH, one of those stress hormones I mentioned earlier on. So basically, they don't return to baseline in terms of their stress hormones after a stress has occurred. They have this longer response. But if you give the reactivation of microglia, the chemogenetic approach, you can rescue that response back to baseline, back to control levels faster than the ELA animals had, which is really cool. So that's shown in the middle graph there. It's also graphed as a decay index on the right side of the screen or the slide. And so that was really interesting. And we wanted to then look at the kind of chronic stress response, which is really kind of nicely indicated by adrenal gland weight. So you can just weigh these adrenal glands and see if they're enlarged, which is often seen with situations of chronic stress. And it's something that we see after ELA in our animals. So in that graph on slide 37, the dark pink bar is showing a significant increase of adrenal weight in ELA mice, but the light pink bar are the animals that got the microglial activation or reactivation. They actually show a return of adrenal weight to normal by this intervention, which is really cool. So we basically prevented the ELA-induced increase in adrenal weights, this kind of chronic stress marker. And then finally, we wanted to kind of look behaviorally at these mice more in depth in their stress response. So we used this looming shadow task, which is pretty interesting, pretty nice behavioral task. So basically, because mice are prey animals, they will react to a predator stimulus in multiple ways. Multiple defensive responses can be seen. So we will often see escape, where the mouse will run away from that predator stimulus. Oftentimes you will also see freezing behavior, where they decide, okay, I don't want to run away, I just want to freeze. That's more of a passive response, so hopefully the predator doesn't see them. But we basically can assess how these animals respond to this, basically a PowerPoint animation of a growing black circle on a screen overhead that looks like a looming shadow of a predator, right? And so what we see in our mice is that the ELA mice actually are slower to respond. So we looked at the latency to respond. We didn't see any changes in how they responded, but we saw like what they chose, escape versus freeze, but we saw that they were slower to respond. And we also saw that if we give that chemogenetic reactivation of microglia in the ELA mice, we can rescue their response rates to the control levels again, so returning them to normal like we saw in the previous graphs. So that was really exciting that functionally this seems to be not only rescuing their synapse number, but also their behavioral response to stress, which is kind of what it's, what's most important here. So finally, moving on to the summary on slide 39, just to sum up what I've told you today. Early life adversity is resulting in unpredictable maternal care, and this results in an inhibition of microglial function, we think likely through MerTK signaling or phagocytic receptor pathway that I discussed. And this inhibits microglial synaptic pruning, which then results in the increased number of excitatory synapses on stress-sensitive neurons in this important brain region for the stress response. This leads next to the altered epigenetic programming of these neural populations, which we know is causing long-term increases in stress hormone release like CRH and CORT. And then this leads to altered stress circuit maturation during development. And finally, the susceptibility to stress-related emotional disorders such as anhedonia and depression. So with that, I'd just like to thank everyone that's contributed to this work along the way. I've had some excellent mentees in my lab as well as in my postdoc lab. Big shout out, obviously, to my mentors along the way, Stacy Bilbo, who helped me fall in love with microglia long ago, and Tally Barham in my postdoc lab, who really guided and sculpted kind of my thinking along the way for this project, and all of our collaborators that made that work possible. And of course, the funding that definitely enables that work to occur as well from both NIMH, the National Institute of Mental Health, and the Brain and Behavior Research Foundation. And with that, I'd be happy to open the floor for any questions you guys have.
Thank you so much. This was so amazing. And yeah, congratulations on the award, because that grant is also an award from
oh, yes, thank you.
the foundation. And it's so cool that you use dreads and stuff in glia. I just use them in in neurons. So
right,
that's
right.
really
They can
cool.
work in other cells. I know.
That is really cool. And yeah, congratulations to your work, and it's really great. I'll give the questions to everyone. I just wanted to ask a question for maybe for the future. Are you looking to also looking into other glia, like we did maternal separation?
Maybe it broke up, Katerina. I heard maternal separation.
Connection issues, yeah. It should get restored in a second.
Okay.
maybe more than a second.
I know.
We need elevator
Well,
music.
I know.
are you there, Katerina, or should I ask a question?
She went
Or
to mute, which is probably indicating that she's having connection issues on her side. So
For me, the
maybe,
icon is gray though, so that might mean that she's either getting a phone call or the phone has failed.
Oh,
So
sorry.
there we go.
Sorry.
Okay,
hey,
back.
Oh,
you're
you're
back.
back.
I'm really sorry. So what did you hear? I was talking and
Just
talking.
start from the
Oh, no
beginning.
worries.
yeah,
Yeah,
basically.
I'm so
Yeah, you
sorry.
cut out at
So
maternal.
congratulations. Oh yeah, we did some maternal separation and we saw in RNA-seq data some differences in the oligodendrocyte, and then
Yeah.
also in like long-term cognition deficits in adults and in habitual behavior.
Oh,
Did you
cool.
maybe do something like that? I know we do way more behavior work than we publish, so I wanted to ask.
Right. Yeah. No, that's a great question. Yeah, and I definitely want to do some RNA sequencing of these microglia. That's the next step with my grant. But I have not looked at oligodendrocytes yet. I think that would be really cool to look at in this model as well. I also haven't looked at habitual behavior, but I am hiring a postdoc who has been looking at operant conditioning type tasks that could be used for that kind of question. So it may be in the cards in the future. Um,
Oh, cool.
but
Yeah, motivation
yeah,
was like really different. Like motivation was
yes,
down. So yeah.
yes, yeah. And we actually see changes in like self-administration of drugs of abuse. So like motivation to take cocaine is way down in these animals. Um, and it's actually very dependent on the sex of the animal, which I didn't get into in the talk at all. But the males, have less motivation to take cocaine and also opiates, but females take far more opiates than controls if they had an ELA experience. So yeah, it's sexually dimorphic,
Oh,
which is
that's so
really
cool.
interesting. Yeah.
Yeah, thank you. Please go ahead. I don't want to take over the stage here, take advantage of my position.
Thanks,
So,
Katerina. Yeah, no worries.
Just quickly
Thank
Serena,
you.
then.
before we get into that, how much longer do we have the honor of your presence?
Oh, well, let's see. I know I was kind of planning for 10, but we can go a little over that if we need to. Like 10, 15, maybe?
Okay, so about 25 minutes. Right on. Thank you.
Yeah.
Yeah. Thank you so much for sharing that. Oh, sorry. Go ahead, doctor. You wanted to say something?
Oh, I can wait. I had a question.
Yeah, I do. I did too. So go ahead, please.
Okay, so that was wonderful and very well delineated. Thank you so much for that, Dr. Bolton. The
Thank
thing that kept
you.
coming up, thank you. Two questions kept coming up into my mind. One was gender related, of course,
Yep.
um, and that branches out into two separate also questions, and one of them being paternal versus maternal. You showed the maternal effects, I guess, more or less,
Yes. Uh-huh.
right? Okay, do you want to talk
Yes.
about that before I continue on?
Great question. I know, because I'll forget the second part. So yeah, it's so interesting because you can't really assess paternal effects so much in mice because maternal care is so dominant in this species, as it is in many mammalian species. Of course, if you look at humans, you can get more into that biparental care question. What are dads doing in response to kind of an impoverished environment, that kind of thing. And back in my postdoc, my advisor Tally Barham had this awesome collaboration with human type studies where they were looking at what happens in humans that have experienced a lot of unpredictability in their early life. Situation, essentially, like more of a chaotic household versus more of a predictable and controlled household. And it's really interesting because we see very similar outcomes in humans that experience unpredictable and kind of chaotic early life environments and these mice that experienced the limited bedding and nesting and this early life adversity that we have imposed upon them. So we see changes in the reward circuitry of the brain that could be what's really underlying these depression-like outcomes in terms of mental health that we see in both the mice and the humans after this type of experience. So yeah, it's definitely hard to pinpoint in the mice whether it's mom or dad, but I think both could definitely have an impact because we're seeing this in the humans for sure.
You know, I did my undergraduate work with Dr. Anthony Grace, who also has done quite a lot of work with
Oh yeah,
schizophrenia. Yeah.
that's
And
awesome.
yeah, and so, but with his work through a different pathway, he's showing um, the propensity for expression of schizophrenia later in life with early life adversity for those who are predisposed, and that comes in the form of hypervigilance, right? So
Right, yes.
interesting. Yeah, there's so much to discuss here, and I don't want to hog up the
Right.
stage, but I will ask you. So that's a whole nother bag of tricks we're going to put aside for now. But when you look at right, when you look at
Yep.
intergenerational trauma,
Oh
what you
yeah.
end up finding, so this is trauma, it's early life adversity, can be early
Yep.
life, it could be intergenerational. In either case, what you find is hypocortisol cortisolity. I always have a hard time saying that.
I know.
with
Yeah.
with increased glucocorticoid receptor sensitivity, right?
Mm-hmm.
So seems to be a bit contradictory, huh? I don't know. What do you think about all that?
Yeah, that's a great question. And I feel like I've seen, just as much on one side of that as I've seen on the other side in the literature. Like some people find hypercortisolemia, some people find hypocortisolemia, so high versus low, in response to early life adversity. And I think it really depends on the type, the duration, the predictability of these early life stressors that you're talking about. So when something is unpredictable and really not controllable, as in the case of this early life limited bedding and nesting that we have, it seems to result in this increase in court long term. But I think if it was more controllable, maybe if it was more predictable, even if your maternal care wasn't changed to be completely fragmented and unpredictable, like we see in this model, it might be a totally different or the opposite outcome. So this is kind of like the stress inoculation literature to some extent that has been seen in other models. We don't really see that as much in our model, but we haven't really looked at females as much in this model. So that could be something that needs to be explored further in terms of the sex difference as well.
Thank you so much. Excellent. Love it. Thank you.
Thank you.
Yeah, about the cortisol, I know it goes away from this, but there was a huge study about Holocaust survivors and
That's
the
exactly the one that I
families
was
that there
thinking
was,
about. Right.
yeah,
Oh, wow.
where there's lower
And
cortisol.
it's
And
from
I
the
was always
glucocorticoid
thinking if that's,
receptors. Yeah.
yeah,
Yeah,
and
that's
I was always
interesting.
thinking if that's not a resilient mechanism because it's from the few that survived this
Right.
thinkable stress, right? So I was
Exactly.
always
They
thinking,
seem to be.
yeah,
That could
exactly.
be
Yeah.
And
pre-selected
it
though,
also goes
because
through the
no,
sorry.
I was just gonna say it could be pre-selected. Like my grandmother was in a concentration camp, and like a lot of people didn't make it out of there, right? So there was an
Right.
like, unfortunate genetic bias, I think, to the survivors to all have a similar kind of response. So
That's a great
Right.
point.
That's very
Never
true.
thought about that.
Yeah,
Also,
that's a
I
really
think
good
that
point.
in a clinical level, I mean, it can be very different because when we are talking about the cortisol, the patient can just deal with the insufficiency, cortical insufficiency, and this is a different situation. So we
Right.
should just monitor for the blood pressure and the level of the sugar, and they have
Right.
I mean, different situation. So it's not that much that cannot be matched that much. It's the result of the science.
Yeah, that's a great point, and I didn't talk about that either in the sense that these animals had an early life adversity, but, we're studying them when they're not, for the most part, when they're not experiencing in the later stress. So it could be totally different. When there's a later stress, we see obviously the prolonged response, like I showed you guys, in the ACTH levels. But, if you're constantly living under this chronic stress, maybe you eventually exhaust your HPA stress response potentially. So, it's definitely dependent on kind of the life history too of the animal or person. So that's a good point.
I know Serena was waiting for your talk for a long time. So Serena, please go ahead.
Well, yeah, no, oh, thank you. It was such a fascinating talk. I have two
Thank you.
questions. So you paint a picture that the mice are naturally born too sensitive to stress, and the microglia's balance or duty is to go prune and reduce that for resilience. What
Right.
It's interesting that it does last throughout life. So in terms of synaptic plasticity,
Mm-hmm.
is that a region of the brain that is that just those there's nothing to bring the synapses back or prune them?
Oh yeah, good question. Very good question. So the reason that they originally chose this time period in these mice, the P2, postnatal day 2 to 10, is because this brain region kind of almost fully develops, or is at least mostly mature by P10. So this brain region is an early developer, whereas if you're talking
Hmm.
about the prefrontal cortex, which is very important for higher cognition and things like inhibition of impulses and things like that, that region develops a lot later. So we would be having to look at a totally different time point, I think, if we wanted to study that brain region, for example. So it's very brain region specific.
Mm-hmm.
And, I don't think necessarily just because the sensitive period has ended, you can't change it. I think, like you said, there's a lot of plasticity, synaptic plasticity, especially throughout the brain into adulthood. And so it's just a matter of, can you get them out of this rut, I guess, that the circuit has developed into, right? And in this case, we haven't tried to enrich their lives in any way, right, to try to rescue them. But that could be effective. So giving them different types of experiences, like environmental enrichment has been shown in other brain regions to be effective, like in the hippocampus. I don't know that it's been studied as much in this brain region, to be honest, but yeah, things like that could be potentially kind of possible, I guess, to re-induce synaptic plasticity later in life, if that makes sense.
It does. Okay, so that's interesting. The other question was in terms of the MerTK administration to the control. Were you able to characterize any difference in the control animals as to increased resilience?
Good question. Yeah, I'm trying to think back now. Go back through my slides. For the MerTK mice, I guess we were kind of mimicking the ELA effect by getting rid of MerTK since it's inhibited in the ELA. So we didn't see resilience. Yeah, we saw more, I guess, more mimicking of the stress sensitivity in the ELA mice in this situation. But if you look at the controls that got the GQ dreads, which you might be thinking of as well, like if you try to activate the microglia in controls to prune more, which they're already pruning, to a certain extent, what was interesting is that you see kind of a diminished stress response. So maybe kind of suggesting resilience to some extent. It's either blunted or it's so it's either not functional or it's towards this resilient phenotype. So that is definitely a possibility in that group. It's a little hard to say because in the behavior, they did have a longer latency to respond to the stressful predator cue, like the ELA mice did. But, you know, that in that context, maybe that is kind of a resilient response. It's hard to say in that interpretation, but yeah, that's a really good question.
Okay, thank you.
Thank you.
If I may, I think it's wonderful research. Like myself, I had some childhood traumatic experiences with my father, spent a lifetime kind of struggling with it. And I'm always
Right.
fascinated by this kind of research because it allows me to kind of reflect on my own experiences and the processes that sometimes go on in my monkey brain. And so I think, like, if there are senators or anyone, I know I said this actually earlier today about another talk, but I think it's important that legislators and policymakers really pay attention to this kind of work because I think it can shift a
Right.
lot of early childhood funding. Like we look at the
Yes.
differences in these outcomes, we can see, like, you know, a dollar in at age 1 can go much further than a dollar at age 10. So earlier interventions helping families
Right.
and so on is something especially significant now. And given the
Yes.
recent attack in the mass shooting over the weekend,
Oh,
that was
yeah.
kind of something that made me reflect on the structure of families and how some families have to travel really far and they have much harder struggles than others. So thank you
Yep.
for sharing this important work.
Thank you. No, that's a really great question. There's actually a very interesting study at UC Irvine while I was there looking at, yeah, supplementing family income early in child development and like how profound of a impact that could have, you know, just I think they were only giving like maybe $200 a month or something, but it was this predictable infusion of this supply, right, of resources into the family's life, and it seemed like it was having kind of an outsized effect, which was really cool on these children's development, brain development, and cognitive development, things like that. So that is a very good point.
I wonder if there would be a possibility of a study where you could make the provider of the baby mice, so like the parents, have a harder time or easier time and see if there was a similar reaction, kind of, oh,
Yeah,
are the children picking up on the parents' stress difficulty levels? Because I think
right.
that's also something that I often thought about growing up, how difficult things were
Yes.
for my mother.
Right. Yeah. And that's something I often talk about in my talks, which I didn't today. But yeah, it's this whole thing about the maternal care, the paternal care. It's so important because infants, like developing babies and of any species, are so reliant on their parents when they're that young that they perceive the world around them through how their parents are perceiving it and reacting to it, right? So if there's a bomb going off outside and your mom is just not worried about it, then you're not going to worry about it, right? So it's like, okay, this is it's very important to kind of think about how the parents are reacting to the environment, because their reaction can, yeah, completely diverge in terms of good versus bad for the offspring. So that's a really good question.
Thank you so much, Dr. Bolton, for that. I want to see if, because Ferris and Dr. Jordan, both of you posted questions in the chat. So I was wondering if you wanted to ask those questions, Ferris, and then Dr. Jordan.
Yeah, hi. Thank you for allowing me to speak here. And it's a really interesting research. I wouldn't say just one study. It's a really nice collection of work that has sort of built on itself, as well as the work of others in the field, and really
Thank
cool
you.
methods as well. Oh yeah, and congrats on the K99. That's not something to to pass on.
Right.
It's like a pace shift, but.
Right.
Concerning this topic, so coming from the clinical world, as a physician researcher, really, so
Okay.
I obviously I tried to grasp as much as I can from the basic science and the animal models that you were using. So I
Right.
had a couple of questions. First one would be for a couple of the steps that you used and had graphs on, especially those that sort of tried to summarize the change over time as well as the effects after, like in the ELA group of mice as well as as compared to the controls. What was the sample
Right.
size for those
Yeah. Good
experiments?
question.
And
I'm
uh,
trying to think
go
back.
ahead, yeah.
No, I'm sorry. For which graphs you're referring to? Typically, our sample size is around 8 pups, mouse pups. And we try to take that from as many different litters as we can to control for litter effects. So it could be just like we were talking about a second ago, one mom's response to this stressor is not the same as another mom's response, right? So we try to represent multiple litters, at least, 5 or so, if not more from that group of 8 that we try to include in most of these assessments. Some of these groups are even 10 in terms of behavioral outcomes and things like that. You have a little more variability. So it can range, but yeah, typically 8 to 10 is the group size for these things.
Okay, okay, that makes sense. And for the models that you use, the mouse models, did you sort of create them yourself, or were they available commercially? I'm just wondering about
Right.
the how I'm trying to imagine how difficult it might be to create something like that consistently, over
That's
time.
a great question.
So,
Yeah.
yeah,
So yeah, they were not commercially available in the final form that I ended up using them, but the components were commercially available. So that made it a little easier for me. You still have to do some extensive mouse breeding to get to what you need, which takes a year or two, depending on how well they do. But yeah, basically I was able to take kind of two different reporter lines, the CRH TD tomato neuron reporter line and the CX3CR1 GFP, which is the microglial reporter line, and cross those together over multiple generations to get what I needed for these experiments. So yeah, I didn't have to reinvent the wheel to the extent that would have made it much longer of a process, but still
yeah.
probably a year of mouse breeding at least.
Well, kudos to you then. That just makes it even more impressive, to be honest. Um,
Oh,
the,
thank you.
the other two last pieces of questions would be relating to sort of the downstream effects and implications of this. So I was wondering if you had that in mind already, or you're planning to do that in a future step of
Right.
or future grant. Um, that um, to try to sort of remove a few confounders, which one of them was brought up, which is the cortisol receptors and how they usually get downregulated sometimes, or upregulated, or just generally
Yeah.
as a sort of a composite, or not a composite really, but sort of a surrogate measure of quote unquote resilience, I guess,
Right.
or what might be considered resilience in some aspect.
Yeah.
So I think that would sort of even drill it down further in terms of creating not just a simple association, but potentially a possible causal link, which
Mm-hmm.
would, I think, be absolutely amazing if that
Right.
can be established, at least in part, because obviously there's like at least 10 criteria, the Hill criteria for causality, but at least I think
Right.
that would make it clearer and remove a bit of ambiguity that is really hard to remove. And then the other
Right.
thing, yeah, and the other
Oh,
thing,
sorry. Didn't want to
oh,
interrupt.
no worries. Go ahead if you'd like. Yeah, go ahead.
Oh, no, yeah. I was just going to say that definitely the next step in my grant proposal is actually deleting glucocorticoid receptor from microglia specifically. So, kind of getting at your question there, mechanistically, we think these microglia are responding to stress hormones from these CRH neurons potentially in the PVN. And so, if we delete this critical receptor, that is, allowing them to sense this stress from the neurons, then, do we prevent all the cascade of ill effects from happening in the first place? So definitely a great idea for sure.
Yeah. And so the last
Yeah.
one is a bit maybe sort of fantastical, I would say, in terms of being not so realistic, I guess, but just throwing it out there just so that I wouldn't regret not, saying it. But essentially, trying to test this in humans,
Mm.
obviously in a very ethical way, not in a very similar way with these animal models, but um, now I'm sure obviously the animal models have been going through very ethical considerations that are extremely
Right.
strenuous and rigorous and all of
Right.
that. But obviously for humans it's going to be a completely different story to try to even test that remotely. So
Right.
are you aware of any sort of a surrogate measure of what you were measuring in these mice? Um, or maybe
Yes, that's a
potentially
good
something that
question.
can be studied observationally, not in a non-interventional way. I think
Right.
that would be super interesting as well. And I haven't really searched the literature around this. I'm aware of a few studies that have tried to get at this link between the early childhood trauma and
Yeah.
the later sort of psychological distress in the many aspects they can take, from anxiety to depression, et cetera,
Right.
later in life. But obviously, these are more mercurial in terms of their associative links.
Mm-hmm.
And they're obviously far from objective as compared to the studies you're doing. So I'm just trying to think if you're aware of anything that can be done in that domain. And thanks again,
Right.
wonderful work.
Thank you. Yeah, no, that's a great question. And so, yeah, it's definitely difficult to look at microglial function in humans currently. You know, there is some PET imaging that's developing that allows you to look at, to some extent, the level of microglial activation, quote unquote, that you see in a living human's brain, which is awesome. But it's very early days, very rough measures at this point. So, you know, really the best hope would be looking kind of post-mortem at some of these things, if we could get our hands on brains of people that experienced early life trauma or adversity, and later, you know, look at how the circuits are kind of wired up, how their neurons synaptic changes are occurring, and even if their microglia look altered later in life. Most likely we wouldn't be getting mini brains from during development, which is when, you know, I was looking at in these mice, but maybe we could look at more of the long-term outcomes in these populations, which would be really cool. So that's a very good question.
Would
Yeah.
maybe section from when people put in epilepsy implants in,
Oh
or
yeah.
would that work maybe?
Yeah, that's a good point, Katerina. Yeah, usually those are in more the limbic regions of the brain, not as much the hypothalamus as far as I know, but that would be really cool if we could even get insight into those other brain regions and see if this is more kind of generalizable as well. Um, Very good point.
Thank you so much, Dr. Bolton. Um, Dr. Jordan, we're going to come to you in a moment. Does anybody else from our team have any questions? I have one, but I want to make sure that everybody gets their questions answered. No? Okay. Dr. Jordan, if you could. Oh, sorry, Katarina, did you
No,
have
I
one?
said go ahead.
Oh, yeah. Dr. Jordan, if you can answer your question really quickly, and if we have time, I might ask mine, but go ahead, please.
Sure. Yeah. No. When I think of sexual dimorphism, I think of at the organismal or organismic level, and when you reference it, I was just curious if you could elaborate what you meant, because I would normally think of brain density or volume, but certainly not at the macrophage level. But maybe I missed something.
Good question. Yeah, there's so many differences between the sexes that we don't really talk about in terms of molecular level. You know, not necessarily basic cell signaling pathways always, but there are some instances of that even. Um, but I was referring more to in our case, we see, for example, the change in excitatory synapse number in males. We see that show up early at P10 at the end of the early life adversity experience, but we don't see it show up in females until later, like 2 weeks later. It's fully there. It's not really there though early on. So they have kind of a different developmental trajectory in terms of how things are changing in response to early life stress. Um, we basically see kind of different stress responses to some extent as well. And then this leads to different behavioral outcomes, as I mentioned. So in the males, we see more depressive like behavior, more like anhedonia, whereas in the females, we don't really see that anhedonia to the same extent, but we see increased susceptibility to drug addiction. So increased self-administration of opiates in female rats, for example. So yeah, kind of the trajectory and the developmental stages can be different between the sexes, if that makes sense.
Yeah. I mean, I just think of morphism as with regard to morphology, shape, and size. So I was
I
confused
see
when
what
you
you're
mentioned
saying. Yeah.
behavior, but sure. Okay. Thank you.
Yeah, that makes sense.
Thank you so much, Dr. Bolton. Yeah, so you said 10:15, so I don't want to ask you any more questions to keep you longer than you have to.
Oh, you're too kind. I could take one more question if
Oh,
you
okay.
really
All right.
want to ask.
Yes, yeah, it was just something because yeah, I'm kind of thinking about if you had the chance or you planning to maybe study successive generations, because
Uh,
I'm an epidemiologist and I'm interested in molecular epidemiology, which kind of deals with the environmental and genetic risk factors for
right,
certain conditions. So in this case, it would be mental conditions. So
right,
how do some of these things affect successive generations maybe after this traumatic event? So, because we're talking also
right.
in humans about generational trauma and all things like that. So, I just want to know if
Yes.
this is something that you have looked at. Yeah, that's
Oh
it.
yeah, great question. Love that question.
Great
+1 for that
question.
question for me.
Plus
Okay.
two.
I know. I'm surprised no one had asked that yet. Actually, that's a common question I get, and it's a great question. I have not looked at it. Is the short answer, and no one in my postdoc lab had looked at it either in this model. But I'm sure someone is doing that. The question would be, does this early life adversity translate to changes in the successive generations? Is there kind of an epigenetic mark that gets passed on across
Yep.
generations that could uh, result in these long-term changes even across generations, which would be really, really crazy, I feel like, if it did. And I kind of hope it doesn't.
Exactly.
But
Yeah.
I know, I'm like, that's scary. Because, our model, I would say, is not necessarily as profound as a trauma. I wouldn't call it a trauma necessarily. It's more of an adversity. It's, changes in your maternal care that you experience as a pup. So it's an adverse condition for development, but it's not major, like let's say the Holocaust or something like we were talking about in humans earlier. So it may not have as, long-term and huge of an impact across generations. But nobody has really done that. So someone needs to do it for sure.
Yeah, I think so. I think it'd be really great and really interesting to study something like that. It would explain a
Yeah,
lot about human condition for sure.
right. Well, and you just reminded me that, you could also try looking at cumulative effects. Like, you don't even have to look at, you could look at, is that original impact still there, even if you don't have any more quote unquote trauma, but also if it builds up over generations too, which would be really crazy and very scary if that happens.
Thank you so, so
Well,
much
also
for
there'd be
answering.
behavioral changes, wouldn't there? So unless you actually are looking at it on the molecular level, you don't know if it's which comes first, the chicken or the egg, right? Because
That's true.
right, because you're now you've led to a behavioral change, which is going to cause a change in the right in the interaction
Yep.
between the maternal and the pups. So
Right, exactly. Maternal care changes do often get passed on kind of experientially. So those pups could grow up to behave similarly, or not necessarily similarly, but have some kind of perturbation like their mothers did. So totally a good point.
Thank you so much for answering my question. I was
Yeah,
so
no
looking
worries.
forward to an answer. I really hope someone looks into it for sure. But
Yes.
yeah, that's it. That's all we got.
I wanted to comment. We had a researcher here, and I'm blanking on the name. He was amazing. From Australia, that talked about how non-coding DNA,
Yeah.
basically RNA activity, was responsible for extinction training. And then we asked a similar question, and he said there's a lab in Israel that is working on trauma-related generational work, and we should invite him and people that work in this type of field together. He would love to come back and have like a roundtable discussion around this.
Oh, nice.
So yeah, if I get to organize it maybe for the fall or something, or the winter, I would
Yeah.
definitely also ask you to come back, because I think it would
Yeah,
be
that sounds
amazing
great.
discussion. Yeah.
Definitely.
They look into non-coding DNA activity, and that maybe these self-replicating DNA that is ancient viral origin that could maybe be participating in this transgenerational
Right.
transmission. So
Yeah.
yeah, it would be cool to have you all here. So I will definitely reach out to you if I, once I get to have time to organize.
Right, of course. No worries, Katerina.
So thank you so much. It was such an amazing talk, and your work is so cool.
Thank you.
Thank you very much.
It's a great
Yeah.
opportunity.
Yeah, it was so great to have the opportunity to get to know you and to get to know your work better and to hear all the questions. Thank you, everyone, for asking all these interesting questions. I really enjoyed the
Yes,
discussion.
thank you. Great questions.
Yeah, and as I said, come back. I'll reach out to you once I organize this. And
Okay.
a lot of grants for you, a lot of non-bureaucratic, please don't have any bureaucratic hurdles, a lot of money from grants.
That would be great.
But you're doing great, so you don't need any luck wishes from me. So anyways, thank you everyone, and thank you, Jessica. And if you like discussions like this and rooms like this, please follow the Science Society, and we will have also other guest speakers coming. So we will have our next room actually on Wednesday. Dr. Bolani, he will come and talk about how gut bacteria associated with different personality traits. And then we'll have on Thursday Dr. Gabeline. He is talking about how he managed to do mitochondria transplantation between living cells. This is like a really important step for rejuvenation. And then we'll have Dr. Ulyin talking about building complexity in biological design spaces. He is at CUNY here in the nanotechnology department. And it will be a really exciting talk. And we'll have on Friday a room about how dogs recognize dog and human emotions, how they are really good at recognizing emotions from other species, which is kind of impressive. And then on Saturday, we have using Dr. Sherry Cheng. She will talk about using molecular orbital-based machine learning, a new type of machine learning, which will be really cool too. So thank you, Jessica. This was amazing. And thank you, everyone.
Thank you. Thank you so much for the
Tina. Have a
invite.
good evening, everyone.
Thanks,
Yes,
everyone.
you too.
Thank you. Okay, bye.
Bye.
I'll close the
Bye.
room
Thank
in
you. Bye.
3, 2, 1. Bye.